Welcome to this ASO Network webinar titled Infrared Spectroscopy for Biological and Biomedical Applications. This webinar has been made possible by Thermo Fisher Scientific, so a big thanks to them for enabling us to gather today and share science together. Our first speaker today is Professor Dr. Werner Mäntele, Professor of Biophysics at Goethe University. His presentation today will introduce the fundamental aspects of protein structure-function correlation and protein analysis by Fourier transform infrared spectroscopy. Our second speaker today is Dr. Suja Sukumaran, Senior Application Scientist at Thermo Fisher Scientific. She will perform demonstrations using an FTIR spectrometer and microscope and present examples of protein analysis. My name is Darius Sulam, and I am very pleased to be your host today. Now, without further ado, please enjoy the presentation of Professor Dr. Werner Mäntele. Werner, thank you very much for being here.
Yeah. Darius, thank you for the invitation. Suja, thanks for the invitation. My short presentation will take you a little bit through the principles of infrared spectroscopy for biological and biomedical application. This is just a real shortcut showing you what's possible, and I will focus on a few examples of what has been done and what can be made out of that. My affiliation is that of a professor of biophysics at Frankfurt University, but I will also appear during the presentation as the CSO of a startup that we founded 6 years ago that tries to get products out of basic research on infrared spectroscopy. So let's see. The infrared spectroscopy is an established technique which is now approximately 200 years old. If you look at history, you see that the infrared was discovered around 1800, the so-called ultra-red part beyond the visible light by William Herschel, a German immigrating to London, and it became gradually a method for molecular analysis, mostly entering chemistry. So until the 70s, it was basically a method used in chemistry labs for identification of compounds and characterization of simple molecules. That the limitation of the technique was always clear. We'll come to that point in a second. But when Fourier transform infrared spectroscopy entered the field in the late 70s, you see that in the center for the mid-infrared, infrared spectroscopy became more interesting for biochemistry and even later on for medicine. That went from the first tabletop devices having a few square meters of optical table area to handheld instruments that are now available since for the last 10 to 15 years. And luckily, a second improval came around the turn of the century, namely around 1995. The first quantum cascade lasers as infrared sources were developed, which was a big step forward because it overcame some limitations of infrared spectroscopy, such as very low power from thermal sources. And meanwhile, the quantum cascade lasers became a reliable infrared source, tunable or in arrays, and we'll see later on an application of that. The question is, why did it take so long before infrared spectroscopy entered biochemistry and biology and medicine? First of all, the spectral range we'll focus on is around 2 micrometer to 20 micrometer, or as IR spectroscopists used to say, 5,000 wave numbers to 500 wave numbers. In that range, we have water as a strong absorber at several points of the spectrum, which makes an application of IR spectroscopy in biology already a bit difficult. We keep in mind that we have photons in the infrared of very low energy, just about around the thermal energy kT. And the problem is thermal sources, so basically heated rods, glow bars, and so on, thermal sources have a low brilliance, so the inherent problem of IR spectroscopy was always that the number of photons was very low. Now, dispersive instruments require long scanning times. So my first infrared spectra that I took as a diploma student in the mid-70s, they took up to hours. You could easily start scanning a spectrum, take a coffee, have a meal, and then come back and you receive the spectrum. That became a lot different and faster in between. And the other problem was at the beginning of IR biospectroscopy, no sampling techniques for biological macromolecules, membranes, or cells were available. Now that has completely changed. We have now, first of all, we have compact infrared instruments, basically working on the Fourier transform principles, interesting sample interfaces like here on the left side, the so-called attenuated total reflection technique with an easy sample access, and those either for very small sample volumes like a drop of blood, as we see later on, or even flow cells for bioprocess analysis. So the sampling side is also simple today and established today. Now, as I said, we have a second sort of infrared sources, namely quantum cascade lasers, powerful, compact, tunable light sources with lots of photons, which eliminate one of the problems of our spectroscopy. And we have either tunable lasers or laser arrays with lots of wavelengths, which make spectroscopy an easy issue. On the same time, sampling techniques have actually focused more on biological samples. The classical sampling is a transmission sample, has a simple optics and flow systems are possible. Path lengths are very short due to absorption of water. Sampling is a bit complicated. Clotting can appear if you have particles in your sample, and it's certainly not for in vivo. Some colleagues apply reflectance techniques where you shine infrared light onto the sample and collect the backscattered light. This is an easy technique, but the penetration depth is unclear and covers only a few micrometers. A technique I already mentioned is attenuated total reflection, ATR, which is the center picture on the lower line. It's small sample volumes that it allows. You can make flow cells from that. The problem is that it works with an evanescent wave, which has a penetration depth of typically shorter than one wavelength, so in the order of 5 micrometers, and this can be limiting. A bit less known, but extremely convenient for biological samples, especially for working in vivo, are photoacoustic techniques, where you use a microphone to detect the sound wave generated by an absorption process. This is established well for gases and can be used for liquids as well. And I will introduce you further down in the lecture to a photothermal technique, which is simple, linear, has a strong penetration depth of about up to 1/10 of a millimeter, and even possibility to do a depth profiling for skin, for example. Okay, sampling techniques are there. The question is, what are we going to see in infrared spectroscopy? And this is now back to very simple principles. Infrared spectroscopy sees changes of dipole moments with nuclear motions. This can reveal changes of bond lengths, bond angles, bond polarization, of protonation reactions, of solvation, and on the formation and breaking of hydrogen bonds. The spectral range we have to consider is from approximately 2 to approximately 20 microns, 5,000 to 500 wave numbers. And within that range, a smaller region like 5 to 10 micrometers is most relevant for biological samples. What are the pros? What are the cons? Infrared spectroscopy uses small photon energies, which means you don't have unwanted photoreaction, you don't have damage, as could, for example, happen with fluorescence spectroscopy. The photon flow is moderate if you design the experiment correctly. You don't have heating of the sample. Sample quantities are very low by now, so that has been an enormous development. And you don't need high purity. And for those that do time-resolved spectroscopy, the intrinsic time resolution of infrared spectroscopy is 1 vibration cycle, meaning a few picoseconds. So lots of applications have moved to high time resolved IR spectroscopy, and people are now in the picosecond and sub-picosecond region. If there are so many advantages, there have to be some disadvantages. First of all, if you look at the scheme of the alpha helix on the right side, infrared spectroscopy does not have selectivity for individual bonds, meaning that contributions from all parts of a protein, for example, will overlap. And second, I mentioned that already, you have a background absorption of water, meaning that you have to design specific sample forms. That can be done. Let's come to proteins, and here especially to the peptide bond. The peptide bond has a number of normal modes, normal vibrations that are well characterized. Their naming is typically amide A and amide B, and those are the NH stretching modes. Then there is a nomenclature which has Roman 1 to Roman 7, and those this numbering determines the vibration type, and among those, the most important one is the amide 1, absorbing around 16 to 1700 wave numbers, and that's an almost pure CO stretching mode. We will see that this is probably an easy way to characterize proteins. This amide 1 mode depends on the geometry and depends thus on the secondary structure. And that information goes back to the 1950s. There's a paper in Nature from 1950, Structure on Synthetic Polypeptides. And this was the first report that the NH group and the CO group of the polypeptide backbone form a structure that can be probed by infrared spectroscopy and that can be used as a sensitive probe for secondary structure. Along that line, we see that the amide 1 mode of the peptide bond is an indicator for CO bond strength, and thus for its involvement in secondary structures. I would like to have a little warning here in that the amide 1 mode is more than just the sum of CO vibrations. You have some coupling, and this coupling can actually modify the position of the and the intensity of the amide 1 mode. So keep in mind, it's not a simple picture that is just addition of CO bond absorbances. But the point is that if you look at a small range in the infrared spectrum from about 1700 to 1600 wave numbers, you have secondary structure elements that absorb differently. You find regions where you have unordered structure, that's on the top. You have regions where you have beta sheet absorbances, alpha helix absorbances, and you can see that these absorbances are slightly different in H2O and D2O. It's clear because there is a hydrogen or deuterium involved there, and that changes vibrational frequencies. So if you analyze that spectral range very carefully, you can actually get an information on secondary structure. Before I go into detail on that, have a look on the top right of this picture. Here you have a specific range around 1610 to 1615 wave numbers, which is characteristic for aggregated proteins or, for example, amyloid fibrils. These are typical values. The positions of these secondary structures, they vary with the length. The problem of analyzing these is that the half width of the mode is large, the modes are partially overlapping, and the assignment of transitions between secondary structures can be difficult because of that. But generally spoken, you have unordered structure, alpha helix and beta sheet in different regions, and you can use that as a fingerprint of secondary structure for the characterization of secondary structure of proteins, for the influence of mutations, which will show up in that range, for protein stability and protein folding, unfolding, and misfolding. It's easy, it's fast, it's a simple routine, but Suja Sukumaran will take you in more details on that. It can be very precise if you look for changes of secondary structure, but the problem is bands are overlapping and hardly separated. And the question is, where does a certain secondary structure begin and where does it end? I won't give you slides and presentations of the analysis of protein secondary structure, but I will pick up one point here, namely the attractive application of direct following of protein folding. This is a possibility where you can determine melting points by infrared spectroscopy, or you can see how folding and unfolding and aggregation or misfolding happen. I will pick out one small protein for that example. It's a small protein. It's called 10-dimistat, about 74 amino acids, 8 kilodalton molecular weight. And this protein normally inhibits the enzyme alpha amylase. It has 3 prolines. They are marked in red here at position 7, 9, and 50. If you do infrared spectroscopy in that special range, 1700 to 1600 wave numbers from this protein, and if you start at moderate temperatures of like 20 centigrades, you see the clear pattern of a beta sheet structure here according to the X-ray structure of the protein, with a part at the higher frequency, a part at lower frequency marked in red here. If you then warm up the protein to about 40, 50, 60, 70, 80 centigrades, you see how the beta sheet pattern disappears and a clear random coil structure appears. The transition is about 80 centigrades. That is fully reversible for the intact protein. So if you cool down, the maximum moves back to the frequency, and the shape of the curve moves back to a clear beta sheet structure. In contrast to the second experiment, where we have replaced the 3 prolines by alanine at these positions, and if you do the same experiment again, it starts with an unfolding, and all of a sudden there is an aggregation here, and this is indicated by the clear pattern for aggregated proteins in the infrared. This experiment is quick. It has a lot of information in it. If you do the heating up with a temperature jump from a laser, the experiment can be done in like milliseconds or microseconds. Okay, at this point, I will switch to biomedical applications and report a few examples of biomedical applications of infrared spectroscopy, namely the analysis of blood by infrared spectroscopy and non-invasive glucose measurement by infrared spectroscopy. We call all that biomedical spectroscopy. There is an enormous potential in that for clinical application, for portable labs, for home use, and I'll guide you through that step by step. First of all, the mid-infrared fingerprint of molecules in body fluids is the key for that. All constituents of blood, urine, saliva, and so on, if they are molecular type, they have a clear infrared pattern, mostly in the range from about 5 to 10 micrometers, so-called fingerprint range, and that pattern can be used for identification and for quantitative characterization, for example, in a sensor. The basic principle is thus that you record an infrared spectrum of a blood sample, for example, urine samples, sweat, saliva sample. The analysis takes 1 to 2 minutes. It's suitable for point of care and bedside applications. The sample quantities are very low, so we end up with a drop of blood, for example, from your fingertip. This method does not require any reagents. It does not require a recalibration procedure because of reagent change. And the amount of potential infectious waste is extremely low, namely a Q-tip to wipe clean the interface. It can use either FTIR instruments, or as we developed it now, it can be used with quantum cascade lasers. and developed to handheld devices. And the present analysis of a drop of blood, for example, can yield up to 12 medical parameters, relevant medical parameters. But with simple modifications of the sample interface, you can also use it for urine samples or dialysis fluid. Finally, the costs are low. It's just the investment on the device, but no consumables anymore. How is that done? First of all, it uses a compact FTIR instrument. We show here just the sample compartment. You see the ATR crystal. The arrow points to the ATR crystal. The size is a few centimeters. Then it needs a carefully designed mathematical procedure and a carefully selected calibration sample group. You choose the best possible reference. In our case, it was the clinical lab of Frankfurt University. In the case of full blood samples, we have based the analysis of blood on about 2,000 reference samples of full blood and plasma. In the case of dialysis fluid or urine, a lot less. The system has an automatic loading, hemolysis, and cleaning system. It's adapted to clinical routines, and because it's intended for medical doctors, it has a clinically relevant, simple user software. What it uses is mathematical procedures for calibration and validation strategies. Those are multivariate analysis, chemometry, even deep learning procedures. And how it works principally is you take a correlation between a clinical reference value of a certain parameter, with values being in the red range, so potentially dangerous, and the normal range here in green, and you plot that versus the parameter derived from infrared spectroscopy. Now, once you've done a calibration this way, you take an unknown sample, project it on here, and you get the clinically relevant value, which, for example, could be the blood glucose value just determined non-invasively. If we follow that line, we can actually state that here at least 8 parameters, in practice like 10 to 12 parameters, can be determined at clinical precision. In all these graphs, you have on the horizontal axis the real concentration, which means the gold standard, the reference from a clinical lab. And on the vertical axis, you have the concentration derived from infrared spectroscopy, and that is extremely precise for glucose, urea, cholesterol, triglycerides, and as we see in the following picture, also for total protein, albumin, hemoglobin, and immunoglobulin. This means that all these parameters are derived within less than a minute for taking the spectrum and about 20 seconds calculation time from a single drop of blood, and they are all at clinical precision. I give you a short list of that in the next picture. Those blood parameters have been investigated in a range that is covering the full clinical need, and the error is within the limits of what is expected for laboratory chemistry, except in the case of immunoglobulin. This is a platform technology for different medical applications. We have tested it in the university clinics in Frankfurt for point-of-care analysis in a project which is called patient blood management. We have a patent on urine measurements with a flow system, basically an ATR system with a flow cell coupled to a urinal for immediate urine analysis. It's used for real-time monitoring of hemodialysis with a flow cell. And last but not least, it is applied for the blood analysis of small laboratory animals, because the technology I present here requires only a drop, whereas clinical laboratories for used for the blood analysis for animals, small animals, they need typically more blood than the small animal can give without suffering. Okay, let's switch here from invasive measurements of body fluids with infrared spectroscopy to real measurements in vivo. And I'll select here an example which is relevant for many, many people in the world, namely for diabetes patients. Just a few numbers at the beginning. In 2019, about 463 million people worldwide live with diabetes, and unfortunately, this number will increase. The International Diabetes Foundation estimates that by 2045, this will be close to 500 million people. It's increasing in all regions of the world, rapidly in the US, rapidly in the Middle East. In Europe, it's a bit less, it's a smaller increase, but this is something that is really, it's actually a global pandemic. If you look in detail, it's much worse than COVID-19, and it's there and it will not go away by itself. What does a diabetes patient typically do? This is well known. Diabetes cannot be cured, but the disease can be managed, which means that a diabetes patient measures the blood glucose several times a day by pricking, typically the finger, as is seen in the top left picture, using an electrochemical sensor built in a test strip. And this sensor fits into a little device. And this device, after 1 or 2 minutes, gives the blood glucose. The problem is that these test strips are costly, especially if you want to do several measurements a day, and they have a limited shelf time. Although there are, meanwhile, some continuously measuring minimally invasive sensors, this is the bottom right picture, which are tiny needles that are going into your tissue and stay there for a week or two. The problem is that too many people worldwide cannot afford those continuous measuring sensors, and thus they have to rely on the test strips. And typically the test measurements done are not frequent enough. The German diabetes patient measure on average about 1.5 times per day and not more. Now, our concept is to use infrared spectroscopy, and the fluid we focus on is not blood, but it is interstitial fluid, skin fluid. It's a liquid surrounding cells and skin and muscle, and it's not a tiny amount. It's actually twice the blood volume typically. You know that liquid, it appears on skin after shallow scratches, or it appears in a modified version in blisters. And it's fairly simple. It's a matrix of water, ions, albumin, glucose, and phosphate. And glucose in interstitial fluid represents about 90% of blood glucose. And if blood glucose changes, the ups and downs of blood glucose show up with very little delay in interstitial fluid. So targeting interstitial fluid in the skin with spectroscopy is a possibility to get a non-invasive glucose measurement done. How are we doing that? First of all, we use the infrared fingerprint of glucose, which you see on the bottom right. These are glucose spectra in water at concentrations relevant for diabetes patients. The lower line in red here is corresponding to 50 milligrams per deciliter, which is hypoglycemic for a diabetes patient. The blue line here, the next line is like 100 milligrams per deciliter, which is normal glucose, and the top line is 500 milligrams per deciliter for hyperglycemic situations, which is also dangerous for diabetes patients. Those peaks arise from CO and OH bonds of the glucose molecule, and they give rise to that pattern. And this pattern is so characteristic that it can be termed a glucose fingerprint in the mid-infrared. We make use of that fingerprint in the infrared and use a laser system here. You have to send infrared light into the tissue. Infrared radiation at 10 micrometers penetrates about 60 to 100 micrometers, tenth of a millimeter. We don't have the chance for transmission measurements, but we can use photothermal or photoacoustic technologies to detect the absorption process here. How is that done? In our setup, we use an internal reflection element here, top left, in gray, And we use an infrared beam, we call it pump beam, from a quantum cascade laser that shines light through this crystal into skin. The beam penetrates to about 100 micrometer in skin. And if you select the glucose-specific wavelengths for this pump beam, glucose molecules in the interstitial fluid of skin are going to absorb the infrared beam. And as a consequence of absorption, they deposit a tiny amount of heat in these layers. Tiny amount of heat means that these layers are warmed up by absorption just by a few millikelvin. The user doesn't feel it. Now, this warming up of the skin layers in that depth shows up at the surface and is transferred into that internal reflection element. What happens in the internal reflection element, this tiny warming up generates a so-called temporary thermal lens. A temporary thermal lens is something that you know from everyday experience. If you drive in summertime on an asphalt road and the sun burns on the asphalt, you see that the air directly in contact with the asphalt has different optical properties. It changes the optics and you may see mirage effects because of that. And that is exactly a thermal lens. And we use a second laser beam, which is called probe beam, which is a simple red laser diode that we send through this thermal lens, and it is deflected on the thermal lens, and the deflection is detected by a position-sensitive photodiode. This deflection is proportional to the absorbed radiation and to the pump laser power, which we can control. And it's dependent on the change of the refractive index Dn with the temperature of the internal reflection element. It's IR detection by visible light. Whenever Goethe appears here with a flash, it means that it's a Goethe University patent, which is now owned by Diamond Tech. Now, in practice, you use a quantum cascade laser. This is actually a view of a tabletop instrument that we use for testing in clinics and for validation. It has a quantum cascade lasers, which is fully tunable. The quantum cascade lasers, this yellow line here, the infrared pump beam, goes into the finger. The finger layers in skin are warmed up. The internal reflection element senses this warming, and you have a red laser diode and a position-sensitive detector here to detect the deflection, and that is used to calculate the glucose concentration. This technology has to be validated. You see me here at the optical bench. My left hand here is put on the sensor, and the skin of the left, of one of the fingers of the left hand is used for non-invasive measurement of glucose. And at my right hand, we do the control measurement with a commercial glucometer used by diabetes patients. And my task here is to modify my blood glucose. The typical case would be that you have to swallow down 75 grams of glucose in 250 to 300 mils of water. I tell you, that's horribly sweet. There are some tastes for that, like strawberry or so. And the glucose uptake is in less than 5 minutes. Because I don't like this modification of my glucose, my personal alternative is I have to eat about 100 grams of assorted Niederegger marzipan in less than 5 minutes. You see that here. That's my 5 minutes portion to modify my blood glucose. And what you see on the bottom left figure here, you see that the green, the reference, and the red, the infrared value, follow closely over the next 1.5 hours, meaning that the non-invasive blood glucose measurement with our sensing device closely follows the reference value. And if you plot the reference value on the next picture here on the horizontal, the infrared value on the vertical, you see a close correlation, which is within the error lines. Now, we've done that with 100 volunteers, about half of them diabetes patients, the other one healthy ones. We subjected them all to a complete test of their glucose over 2 to 3 hours, ending up in like 2,000 data pairs. And those scatter, ideally, they would be on this line here. the central line, but they scatter because the reference is wrong as well, and we have an error in auto-determination. But we see that 99% of these data pairs are in zones A and B, which are considered safe by diabetologists. So yes, it's possible to use infrared spectroscopy for blood glucose measurement without any invasive procedure. And yes, that's precise enough to be used by diabetes patients. Its accuracy corresponds to commercially available glucometers. That has been published here this year in Journal of Diabetes Science and Technology. Okay, I'll come to an end here. Diamondtech has started with a tabletop device containing a quantum cascade laser, commercially one. This shoebox-sized device can be used by multi-users that identify themselves with an RFID chip and measure their glucose, so it can be used in clinics. A pocket device is on its way. It will come on the market by mid-2022. And a bent type with further miniaturization of quantum cascade laser technology will be available at about 2024, 2025. You see details on that on the Diamondtech homepage down here. Okay, this developments in infrared spectroscopy, in my special case, went over like 40 years. One slide is not enough to show all those that contributed, but I have a few members here of my team at Frankfurt University and at Goethe University and at Diamondtech. And you see the top line are Diamondtech co-workers. The second and third and fourth lines are some of the co-workers at Frankfurt University. And last but not least, Dr. Suja Sukumaran, who will give the next lecture. Thank you.
Fantastic presentation, Professor Mäntele. Wasn't it a great introduction to protein analysis and the next generation of biomedical devices based on IR spectroscopy? I thought so. So what am I going to talk to you about today is experimental aspects of protein analysis. In my agenda here today is to show you a quick demonstration of how to analyze proteins in FTIR, as well as how to use the protein analysis for some applications into food technology and research. When we think about protein structure, stability, and aggregation, and analyzing samples in FTIR, there's two key sampling techniques that we have to remember. One is transmission, for which we can use transmissional cells like these, which are very small path lengths, like 6 to 8 microns, and we can use protein solutions that are in H2O or D2O based buffers. But what we will do today is look at ATR, and we will use both multi-bounce ATR as well as single bounce ATR. So multi-bounce ATR as well as the single bounce ATRs are one of the more popular methods for analysis nowadays because of ease of use, because of ease of cleaning, as well as the opportunity to retain the sample after the experiment is completed. So let's analyze some proteins dissolved in water-based buffers. What we are going to do is use the IS50 FTIR to do our measurements for today. So what you can see in the main sample compartment, I have my multi-bounce ATR that is in the main sample compartment. This is a 10 bounce diamond ATR that is in the main sample compartment. There is a silicon version of it as well that can be used. I also have up on the right side, you can see a single bounce diamond ATR, which is great for powders. So what we will do is we will run some protein liquids with our concentrate IR, as well as we will run some powders with our single bounce ATR. So just to give a quick overview on my instrument just by itself, on the right side, I have this connected to the continuum microscope as well. And on the left side, what you're looking at is it is connected to the IS-50 near-infrared module. But we are going to focus on the main sample compartment as well as on the built-in FTIR for today. So let's set up the experiment for doing a measurement with the concentration. In this situation, first we want to set up our experiment. So a couple of key questions that comes up is which detector to use. So here is where you may want to use an MCT detector, especially if you are working with proteins at very low concentration. Because MCT detector, which is a liquid nitrogen cooled detector, is much more sensitive than the DTGS detector and much more faster as well. So there is option for me to use the MCT and DTGS, and in this case, I'm using the MCT detector. The Collect tab will tell where we can set the parameters for the number of scans and the resolution. Two other key parameters where it is based on what samples you're measuring and what end goal you have. So if you are considering samples that are very dilute, you definitely want to go much higher in scan numbers. The resolution parameter is important because if you are planning to do secondary structure evaluation or look at peak shifts and peaks which are very close to each other, and you want to resolve them properly, you may want to use a resolution of 2. For general use, you can also use a resolution of 4. As you change the resolution, the higher resolutions like 2 and 1 and so forth, the time that it is required to make a collection will be higher as well. And the number of scans is also critical here because there will be experiments where you are looking for very small differences between the same protein sample. You definitely want to go higher in the number of scans so that your signal to noise is really good. So let's start with a background. So in the Omnic software, what you can do is just click on this button that says click background and it will start the background measurement. So in the background measurement right now, what you can see is that I am measuring the background on an empty diamond crystal just by itself. So my background measurement is completed. So I'm ready for either my sample or the buffer measurements just by itself. So all I need is 10 microliters of my buffer or the sample. So here I will go ahead and load 10 microliters of my buffer solution. So I just loaded 10 microliters of the buffer solution in there, and I'm ready to start my measurement. All I need to do here is click collect sample, and I'm ready to measure. And so now we have completed the measurement of buffer and protein. The next step would be to subtract the buffer. So to subtract the buffer, all we have to do is click on the select all button and then under process, click on subtract. See clear amide 1 and amide 2 region as well as the amide 3 regions. So now we have to use the amide 1 region for our peak resolution or for peak fitting to get the secondary structure. So for that, we just select the amide 1 region, and all we have to do is go to the Analyze button and click on Peak Resolve. In Peak Resolve, once we have selected the peaks and we have described the noise levels, all we need to do here is to click on the Fit Peaks button. This will run quite a few iterations to fit the peaks into the spectrum by itself. So once the peak fitting is done, all we need to do is click on the button Add, so that it just adds it into a new window. We can also look at the peaks and the peak areas as well by just looking at the peaks just by itself. So you can start to see the various peak areas that the software has come up with. You can add and delete certain peaks as well as required. How to analyze the proteins using a multi-bounce FTIR. So let's just summarize what we just learned. So when we want to analyze proteins that are in water-based buffers, we need to collect a buffer spectrum. We need to collect the protein spectrum, especially if we are going to do this at different temperatures or so. We definitely will have to collect the buffers at different temperatures as well as the protein at different temperatures. Then we subtract the buffer from the protein spectrum, and then the subtracted protein spectra with the amide 1 region between 1700 to 1600 is used for the peak resolve or the peak fitting function. And more information can be obtained under the Omnic training videos on our website. But there is a second way to analyze this, and that is using the BioTools Proda3S software. So using the software, you can do multiple spectra at the same time, but this software is also more geared towards transmission analysis. So if you have samples where you are doing multiple samples in transmission, you can use the Proda3S software for analysis of secondary structure as well. Now let's switch gears a little bit and talk about the applicability of what we have just learned. So one of the common place where we can think about applying what we just learned about protein secondary structure determination is when we think about powdered food. So for simple things like whey, rice, milk protein, pea protein, all these materials are complex mixtures of carbohydrates, proteins, and lipids. But to analyze them, it's super simple. So we will analyze it in a single bounce FTIR. In the next 2 slides, once we look at the data, what we are going to look at is how powerful it is to use a PCA-based analysis of these powders to basically use it for classification and discrimination analysis. So let's analyze some milk powder, a simple milk powder sample. So I have the sample right up here. All I need to do is take a small scoop of it and put it on the FTIR crystal. So you can also see on the screen, I have already completed the background. Once you have a powdered sample or so, it is important to use the pressure tower that is right in there so that the sample is in proper contact with the FTIR crystal by itself. Now all I need to do is click on collect sample and it just starts its collection. We just learned how to analyze powdered samples. So now let's look at some results from some powdered samples, just like what we just measured. So in this slide, what we are looking at is milk protein, pea protein, rice protein, and whey protein from different vendors, as well as from different lots that were analyzed, and they were analyzed repeatedly. But what was observed is that even the same type of protein from different vendors showed subtle differences. Then these spectrum were they're further processed using PCA analysis, and second derivative of these spectra were used for such analysis. When such an analysis was done, what we observed is that each of the protein would separate out into its own little principal component regions. So what's the advantage here? So the next time a certain type of protein sample from a particular vendor arrives, they can compare it to this particular model and say, oh, so this one is much closer to the G1 group, or this is particularly very similar to the milk protein group that we saw in the A2 type, or so forth. And learn about the Rheo-IR. A Rheo-IR is an instrument where rheology is coupled with FTIR spectroscopy. So what does rheology in general do for us when we think about food or beverage by itself? Well, we want to know about viscosity, and we also want to know about the elasticity of material. So we want to know about the viscosity of things like chocolate syrup, and also elasticity of materials like marshmallows. So how do we couple two of techniques, one studying the physical properties of materials like food powders, especially proteins, and then couple it with the FTIR spectroscopy. And that resulted in the instrument called the Rheo-IR. So let's look a little bit deeper into the Rheo-IR by itself. So what are we looking at here is the schematic of the Rheo-IR on the left-hand side and the Rheo-IR in our lab on the right-hand side. So if we look at the Rheo-IR, it is composed of three main components. One is the FTIR, the other one is the Hucke-Marsh rheometer by itself, and then there is the interface between the FTIR and the rheometer. And this interface, the key component is the diamond ATR, which is built under the steel blade of the rheometer by itself. We will take a closer look at how we load sample into the steel plate, as well as we will look a closer look at the ATR by itself. So we are loading the sample for the rheometer. So you can see the whey protein is being loaded right into the steel pedestal. So underneath there is also the diamond ATR crystal. So the RheoWin software controls the temperature ramps, but it also talks with the Omni software. So what we are doing is simultaneous collection of the rheological property as well as the changes in the FTIR peaks using the FTIR by itself. So the temperatures are controlled here right on the pedestal when the plates make the contact. So let's look at the results by itself now that the temperature ramp up and cool down is over. So what happens is, as shown in this picture, at the end of the experiment, the liquid material that we loaded in has become into this nice gel-like material that is also flexible and can fold and so forth. But at the same time, what happened to the protein by itself? If we look at the FTIR spectrum, what we learn is that there is significant changes in the amide 1 and 2 peaks, which is indicating that there is some aggregation of protein that has happened, as there is a downshift in the peak position, as well as there is change in the intensity of the material. So what did we learn from this study? So basically, whey protein, when you actually start off in the solution state, it is in a nice folded state. It unfolds as the temperature ramps up, and it results in an aggregation, and the whole gelation process happens. So Now let's summarize what we learned for the last 15 minutes. Well, we learned that we can use protein secondary structure, folding, and aggregation studies, and we can use it for a lot of different types of application, as well as we can do these using transmission, and then we can do this with ATR, we can do this with single bounce and multi-bounce ATR. We also learned that we can use the power of FTIR spectroscopy and combine it with PCA analysis to create some really powerful discriminate models to do some nice QC techniques. We can also use the hyphenated techniques like the Rheo-IR, which we just did, as well as other things that like the TGA-IR or GC-IR to understand various physical and chemical properties of everyday materials. So with that, thank you very much for joining us today, and we look forward to some questions.
Great. Thank you very much for your presentations and your demonstrations today. I'm going to be bringing Professor Dr. Werner Mäntele back online here, as well as Dr. Suja Sukumaran. Hello, everyone. We're going to jump right into the question and answer period here, because we have a number of questions from our audience that have come through. We do not have too much time for the whole group of questions, but we will try and answer a few here. So the first question I have here is, this one's directed to you, Werner. You mentioned FTIR spectroscopy and quantum cascade lasers. What is the main advantage of using quantum cascade lasers?
The main advantage of a quantum cascade laser is the high power at a distinct wavelength. So in the case of a Fourier transform instrument, you have a thermal source having a few nanowatts per wavelength interval as measuring light intensity. A quantum cascade laser easily has a few milliwatts, so you can get to better signal-noise ratio in this case. On the other side, a quantum cascade laser is either single wavelength, or if it's tunable with an external cavity, covers only a relatively small spectral range, actually up to about 300 wave numbers. So quantum cascade lasers are not yet available as full range spectrometers, but they are available for dedicated tasks in a certain smaller wavelength range, and their intensity is a real advantage to reach a better signal-to-noise ratio.
Great, thank you so much for that answer. Here's another question for you, and this one comes from one of the audience members who says, I use IR spectroscopy a lot for chemical analysis. Normally I use the routine KBR pellet and sampling technique. Can I also use this standard technique to analyze protein samples?
I would not recommend that. The reason is that for simple molecules in chemistry, they can be embedded in a KBR matrix and still keep their normal properties. If you now take a protein in its dry form and put it into a potassium bromide matrix, you have to expect that the outer layers of the protein are heavily perturbed by the matrix, and you would not get a reasonable results to be compared with in vivo properties of the protein. So for the spectroscopy of proteins, you have to either go to the ATR techniques in liquids, as Suja Sukumaran has presented them, or you go to transmission cells with concentrated solutions, and you'll get results that correspond to the native protein as you find it in cells, for example.
Great, thank you so much. I think we have time for just one last question here. And this one comes from another audience member. Can we use peak fitting in IR to monitor protein aggregation and quantify the aggregation level?
I think that's directed to Dr. Sukumaran.
Yeah, I will take on that one, Darius. Yes, so the answer is you can do peak fitting and you can quantify the different forms of your secondary structure. So whether it's the beta sheath, alpha helix, or the amount of aggregates that are in there. Yes.
Great. I think we'll leave it at that since we are just over time here. Thank you so much to both of you for answering these questions and for your great answers and providing us with this the insight in your presentations today.
Absolutely. Thank you so much for joining us, all of you. It's been a great pleasure.
In closing, thank you again for taking part in this Thermo Fisher scientific webinar supported by the AZO network, and we look forward to sharing science again with you in the near future. Thank you.